Well, this is the third and final portion of our lectures on replication. In the last two lectures, the first one we talked about how we figured out it was semi-conservative replication and what that meant. In the last one, we actually walked through all four of the major stages of replication using a kind of a combination of bacterial information as well as eukaryotic information, mixing those together. Our purpose, again, is not to confuse you about that. It's to simply point out there's some shared commonalities, and what we know about it has these major points. These things have to happen for replication to occur. It may turn out by the time you finish college that we've renamed some of the parts, but certainly we've talked about the primary enzymes, or at least some form of what needs to be there in order for it to work. Okay? All right, so we went through initiation and the complexing of that and licensing. We talked about unwinding, how helicase comes along and breaks apart those hydrogen bonds to help us form replication forks and all the other processes that go there, including gyrase. We talked about putting an RNA primer down to get the process started, and then finally the elongation, which involved both a leading and lagging strand. Now what we want to do now is talk about the ends of the chromosomes. It turns out that when we go through this process of replication, we leave some parts at the end, particularly in eukaryotic chromosomes. Obviously, we're talking about not circular ones, but regular ones. We leave a gap, okay? It happens at the end of every chromosome, both ends, okay? And there's always a gap, and there's a gap because there's not going to be a primer that's being put in place there, okay? And so what has to happen is that we have to somehow fill those gaps, and those gaps are very, very important. We've already talked about in an earlier lecture that these telomeres are, each time we go through replication, we get a little shorter, okay? And keeping those telomeres in place and keeping those ends of those chromosomes sealed up is very important, right? One of the reasons for that is because in our cells, we're constantly being invaded by new types of viruses and other things that attack and try to break down DNA, right? So we're being attacked. We're trying to defend ourselves by breaking up RNA and DNA. If you just have naked DNA sitting here with no telomeres, these things are just eaten apart, okay? Our own body will attack our own DNA. It's one of those odd things. So one of the ways we protect that is by having these telomeres, which also serve as caps on the end of our chromosomes, okay? The enzyme that's involved in this process is known as telomerase, which again is it's a complex of proteins and other things. It's actually what's known as a riboproteinase. It's got some RNA components into it and all sorts of things, so it's a complex, but we'll just call it telomerase, okay? Now, just from a very, very cartooned view of what's going to go on here, we've basically got these ends that are left unfilled. Okay, we were never able to put parts down there, and we've got to fill them, okay? And simply telomerase comes in, locates those areas, and adds the bases that are necessary to complete the process, okay? All right, now, these telomeres have some characteristics. In humans, there's a long repeat sequence of TTA-GGG, 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 repeated hundreds of times. In fact, it can be many, many hundreds of times, hundreds to 1,000 different kilobases involved here. So we can have 1,000 of these bases long or whatever, okay? Now, each time we go through replication, each time those skin cells down here at the base are replicating, we lose a huge chunk of these telomeres, okay? We will lose sometimes 25, 30,000 bases down through this region. So they do significantly become shorter and shorter and shorter through each replication. And again, we talked about that being the Hayfield prediction, the Hayfield phenomenon, that as cells get older, that we can tell because the chromosomes are getting shorter and shorter. At some point, that leads to sort of this idea of senescence, that the cells just get so short, that at least the chromosomes get so short, it signals the cells to die. All right, not only we fill in the ends down here, but we also have to put caps. So there's actually some backfolding of this DNA to try to make essentially a sort of a fancy cap that comes into play and kind of molds back around like this. If I can get a color here that will let us see it. There we go, something like that probably would work. We actually get a cap that folds back like this at the end and makes something like that, so that you get a little cross bonding going on here, so that when anything tries to attack that, as far as any kind of enzymes that come along, they're not able to easily grab a hold of this, right? So these overhang caps are very important. Just to take us off for a moment, adipose, the fat, particularly the fat that brown fat we have around our bellies and things, the white fat of that, brown. In mammals, it actually can kind of take over, and it's the process of using P53, which is a protein, produces a ROS, which is a reactive oxidative species. And what that reactive oxidative species does, this particular one, is it sends it out as a signal to other parts of the body to say, hey, we need to collect fat. The organ of fat becomes an organ that says, hey, I want more of this. And the reason it does that, of course, is that in natural systems, when food wasn't so readily available, you had periods of time when there was a lot of food, and then you had winters and stuff where there was very little food. So when there was food, say in the fall of the year and there was lots of food, fat would start to deposit, and it would tell the system, man, you better get more of this, right? Well, now, of course, we have food all year, so it becomes a problem. Where this is a real issue is that in morbidly obese people, particularly those that have lots of belly fat, you're sending out these signals, and those signals are affecting the telomerase capability. And what it does is it means that telomerase isn't working as well because of this oxidative problem. The DNA is getting shorter and shorter in each replication, so people that are morbidly obese actually age much quicker than people that are leaner. It's one of those weird things. It's because adipose is kind of taking over and causing poisoning the other parts of the system. All right. Okay, now, this is one of those where I'm going to show you and never expect you to understand all of it, okay? I'm simply trying to show you that silly little diagram we had a moment ago of kind of a primer or telomerase coming in and sitting down and throwing a few bases on is nowhere near the answer, okay? Particularly in eukaryotic systems where we're dealing with here, it's quite complicated. We have to have little loops that form DNA structures. We have to have an RNA primers that come in. There's all these different poles. There's all this weird chromatin structure, and there's even a loop that occurs back here. Okay, this D-loop back, and in order for that fork to work to make these telomeres. So it's a very complicated model. We're just now starting to understand even the pieces, and this is just a proposed model of how those pieces fit together and where they work. Now you can imagine that most of these pieces are either pieces of RNA or pieces of their proteins. And if you have a mutation that affects the POT1 or something like that back here in another piece of DNA, you may not get the right telomere formation, and you get premature aging. You get all sorts of things that can occur, these TRAPs and others, or mutations that don't show up as being wrinkled wing or straight wing or whatever, show up in these areas, can still cause major issues in the system. But anyway, I just wanted to show you the complexity of why I said we don't really understand this full process. We're starting to work on it, but we do know a lot about the components that are there, and we're starting to guess at how those components fit together. All right? Now, let's take home message from this whole process. The whole process is that you've got an old strand and a new strand, old strand and a new strand. And what you've done is you have finally come along and you have taken this piece of DNA that we had earlier, right here, somewhere here, and we've taken that piece of DNA, it's got a little centromere, right? And now what we have done is that's double-stranded DNA. We've separated that out, and we've made this thing called a sister chromatid right here. So now we've got this thing, we call it a chromosome, but in reality it's called a dyad, because of di meaning two parts, okay? And that part does not occur until we get after synthesis, right? So one of those first days when I asked you, draw a chromosome, and everybody came up with, well, yeah, I can do that. This chromosome looks like this, and I said, almost never looks like that. Okay, most DNA looks, something like this, and only after replication do you start to look like this. Now, even after replication, it doesn't look like this, it doesn't look like this nice little dense chromosome thing that you like to draw like this, and I certainly like to draw it. That's how I learned it. Okay. It doesn't look like that at all. That's after condensation. That's not until we're in the meiosis or mitosis. What it looks like after replication is long, strands of DNA with its buddy over here all kind of next to it, all lined out. But it's not, certainly not in this compressed form. All of G2 is spent getting it in that compressed form that looks something like that. Okay? All right. But from our point, we've now got sister chromatids. Very important for the next few lectures. All right, so let's throw all this together. Just kind of one last throw together of what's going on, okay? What's going to happen? Gyrase relaxes the DNA. How does it do it? It does it by nicking the DNA itself, okay, and letting it spin, all right? Helicase unwinds the DNA. How does it do that? It does it by breaking these hydrogen bonds in here. And again, all these gyrase, the topoisomerase of gyrase, helicase, they're complexes, right? But we're just going to give them a single name now to help us understand the process, okay? You have to have primers put down. Those primers are put down by primase, okay? All right? Once those primers are down and primases come into play, then DNA pole can come in. And DNA pole works because it only works in one direction, 5 to 3 prime here, reading 3 to 5. That means you get one strand going one way, that's the leading strand, and then one strand that's very choppy going the other way with lots of different primers in it, and that's called the lagging strand, okay? Leading and lagging strand. Now, it doesn't have to be on top or bottom. Cells don't have top or bottom, okay? Don't get confused. Just look at the ways. One of the things you always have to do to answer these kind of replication problems is look at these ends and see what we're talking about. Which one's the 5 prime, which one's the 3 prime? Is it down here, is it up there? You need to know where those are, and once you figure those out, then you can come back and locate it, okay? All right. DNA pole, when it hits a primer, it removes it, okay? And then, of course, we need that last little fill-in, which is done with this enzyme the complex we call ligase, right? All that occurs. If I were to throw this drawing up on a thing, those were the pieces you'd be pointing to, all right? Okay. And then kind of the last thing, just to remind you that most of the stuff in Kornberg's work and other things came from prokaryotic systems. But it lets us extend over into eukaryotic systems because as we learn more about it, we can just replace the ideas but with the names of the particular enzymes and things. But the process is basically the same, okay? Prokaryotes tend to have large circular plasmic DNA. Eukaryotes have chromosomes, these long things. We have one origin usually in a prokaryotic system. In a eukaryotic system, we have thousands of origins along the way. Okay, the DNA that's found in prokaryotes there's no nuclear membrane for the most part of any form that we would think of, and so the DNA is just floating around, available to be worked on. Inside of a eukaryotic, we're going to find out that the DNA is in the nucleus, and not only is it in the nucleus, but it's packed down in the nucleus in various ways, and you have to move all that packaging off the outside of it in order to work it. But other than that, it's still very similar. With the exception of things like the telomeres and telomerase and all that, there's still, primase, there's got to be something to prime. You can call it primase for now. There's got to be something that's going to grow the DNA. We're calling those DNA pole. There has to be a helicase to open it up, and there has to be some kind of topoisomerase or thing we're calling gyrase to make the process work. Okay? All right, so what should you know? Well, obviously you need to know all these enzymes, not the big piles of them that are put out there, but the big ones, right? You need to know the licensing factors and the things that are involved there. You need to know that you've got gyrase, you've got helicase, that you've got primase, you've got DNA polymerases, you've got ligases going on, you've got telomerase. Know all of those, okay? Know what a replicon is. That's a site of replication that we have, okay? Realize that replication only occurs in single-stranded DNA, so somehow you must rule that apart, okay? Once you pull it apart, it only works in one direction. Okay, 5 prime toward 3 prime, reading 3 to 5, writing 5 to 3. That means that you're going to have this bidirectional going on, but you're going to end up with a leading strand, either the top or the bottom, and a lagging strand, right, depending on which side you're on. And of course, it's semi-conservative. We've learned that from earlier. As you pull it apart, you keep an old strand, you get a new strand in place, old strand over here, new strand in place, and you end up with semi-conservative replication. All right. Now, the biggie for us, we just back up for a second, is this, okay? We've now created sister chromatids, right? That's and very important for us, this happens during S phase. It doesn't have anything to do with meiosis or mitosis directly. It needs to occur as a precursor to it, Certainly cell signaling, this has to happen before you even will start meiosis or mitosis, but it's not involved in it. It's not part of meiosis or mitosis, okay? It happens in a completely different phase that's out here. All right, well that completes what we want to talk about as far as replication. I think we've got enough of the bases there. Again, it's very cartoonish, I understand, but this is, sort of entry genetics. We're just trying to get the model out there of what all the pieces have to be and then what the end product is. All right, so now we've got that. We'll go and we'll be able to take that and we're going to move to a completely different thing and start talking a little bit about Mendel. And then we'll come back to meiosis, which involves this. So we link Mendel with meiosis, but to do that, we have to understand about replication. That's why we spend the time doing this, okay?